Yeah, so it's a pleasure to welcome you all back after the break, and unfortunately, still in a virtual version of the seminar series. But the advantage of that is it's a lot easier when the temperature outside is -25 to get to the seminar. So it's my it's a great pleasure today to have as our speaker one of our own QLS supervisors. And just as a quick aside comment, yes, audio is working, I think, for everybody else, Kiri. So we have Dr. Junding with us today, who arrived quite recently at McGill. And he is in the Meakins Research Institute in the Department of Medicine. He's an assistant professor there. And before that, he was a postdoctoral fellow in computational biology, School of Computer Science at Carnegie Mellon, and received his PhD in 2016 from the University of Central Florida. He has been doing a lot of work in single cell analysis of single cell data and the single cell technologies, perhaps particularly focused on cancer. And we are really looking forward to his presentation today. I will say that even though he hasn't been at McGill very long, there has been a lot of interest among the QLS students in interacting with him and in working with him. And so it's a real pleasure to have him as part of our team or our supervisor body. So please go ahead, June, and looking forward to hearing your presentation.
Yes, thank you for the very nice introduction. So can you see my screen now?
We do. We see it with the slides on the side, though.
Presenting mode. Is better
there you
now?
Go. Yep.
All right. Hello everyone. And today I want to talk about my, recent studies in single cell genomics. To be more accurate, I want to study, this talk is about the decoding the cellular dynamics from single cell genomics for more effective self-feed manipulation. And here is some concept that I want to elaborate a bit more is what is cellular dynamics? And here it means, the cell state change over time or over space. But in this talk, we only focus on the temporal cellular dynamics. And the goal is, from the single cell genomics dataset, we want to get a better understanding about what drives the change of cellular state and how we can better manipulate the cell state. So, and before going to the actual, the method part, computational models, I want to briefly talk about, cellular states and single cell genomics, as a background. And here we know the cells as actually, cells are the basic building blocks, the fundamental units for almost all life forms, excluding the virus. But surprisingly, we actually have relatively limited understanding about all the cells. We know there's 37 trillion cells in human body, but we don't even have a accurate number of how many cell types are there in our body. We know there's around a bit over 200 cell types, but we don't know actually where are the cells located at, in what tissue and what kind of markers those cells are expressing, and also the functions for each of these cell types, right? And not mention the existence of many subpopulations, right? You talk about T cells, there's different type of T cells, macrophages, different type of macrophages, and there's, is always, discovering new cell types, new subpopulations in a lot of studies, like in the cancer studies, in the cell differentiation studies. So, but, actually, we always want to understand, have a better understanding of all cells in our body. And, is the reason for that is a lot of disease was caused by, fundamentally speaking, abnormal cellular state. Like you're taking the cancer as the example, right? So the cancer, was caused by, of course, abnormal cells. The some cancer cells, they are, keep proliferating themselves and keep invading other cells. Of course, if we want to find or therapeutics against this cancer disease, first thing is we need to find out what other abnormal cells. But to do that, you need to know, to find these abnormal cells or abnormal cellular state, we first need to know what are the normal, right? In that sense, we need to define or, build or a comprehensive searchable Google map for the cells in the body. And you imagine we have 37 trillion cells in the body. So it's gonna be a lot of work, to build such or reference map. And it definitely cannot be done by a single group or, a few people. So, and recently we have a lot of initiatives to build such comprehensive reference of all cells in the body. And the most famous initiatives include, like, for example, Human Cell Atlas or, HubMap. They all try to build a comprehensive reference map for all the cells. But, to achieve this goal, one technology we have to use is single cell genomics, to profile the cells at the individual cellular state at the level of the individual cells. And I want to briefly talk about what are the single cell sequencing and how it differs to the conventional bulk measurements or bulk sequencing. So in here, I was using an analogy here. It's like, the conventional bulk sequencing, like the bulk RNA-seq, is like we're getting a smoothie, right? Imagine that the sample have a million of cells. The RNA-seq readout can only give us one set of readout, with the average expression for all cells in the sample, like a million cells in the sample. So like, always this averaging thing in many cases is really, misleading, and even not just inaccurate, but misleading. For example, if I average my salary with Bill Gates, I will be a billionaire, but in reality, I'm not, it's not even close to that. So this average type of thing is not or very accurate, especially if we use, if we will study or very heterogeneous population, heterogeneous sample like in cancer, and averaging all signals will be very inaccurate.
Julie, I did ask you at the beginning, do you want to wait for questions at the end, or do you want to, how do you feel about questions in the middle? We have one question in the one raised hand right now from Julian Willett.
So yeah, I would prefer like, more interactions along the way. So if you just ask your questions.
Go ahead, Julian.
Okay, can you hear me all right?
Yes, absolutely.
Oh, okay, so I guess so. What I'm hearing you saying, when I'm hearing you talking about having some sort of initiative to type everything, I'm drawing parallels to where this could you could arguably have a similar project for typing cells. Do you think single cell investigation is advanced enough in order to have a more definitive assay, like a more definitive test at this point? Or do you think further things need to be known, so like we only have to do it once per se?
So yeah, that's a very good question. So I would say single cell is still, although we have developed many years now, almost a decade, but it's still not very mature, to define the cellular state. In many cases, the RNA-seq, which is the most popular single cell technique right now, is not enough, So, but other single cell, like single cell epigenetics, single cell proteomics, because if we want to define the cellular state, we need, to describe the cellular state from multiple perspectives. But others, the other single cell measurement are still very, at its infancy. In other words, I don't believe the current single cell measurement can, depict the cellular state very accurately. So we still have a long way to go to build a really, useful, accurate, comprehensive, searchable Google map for the cells. It that answers your question, or?
Yeah, that's perfect. Thank you.
Yeah. So, but so on the other hand, the single cell compared to the bulk measurements is like, it's like we're getting or salad versus or smoothie. Now, we have a measurement for expression in individual cells, like in this case, right? So we have a high resolution of this measurement. Now we, from the left side, the bulk measurements, based on the color, you probably can guess there's still some information. Like here, you can guess, okay, let's look a little bit pinkish, then there might be some strawberries, cranberries, but you never know the truth. But on the other hand, if we have single cell, then we'll be able to not only tell what fruits are included, but also, the quantity, number of the different fruits. So in the real expression, then it's like we're not only able to depict, to find all the cell type, but also, the cells for each of the cell populations. So it really have the capability to deconvolve the cellular heterogeneity in a sample. And because of this advantage, this technology is developing really rapidly in the past decade. If you look at, here is the the scalability, how many cells we can measure in one experiment. If you look at the scales here, in 10 years ago, we can only measure the expression for less than 10 cells for one experiment. And nowadays we can easily do over 1 million, but usually people won't do that. So the normal size of single cell will be a few, maybe 20,000 around that level. But we are able to do 1 million if you have enough money, even more. So, but as I mentioned, not just the single cell RNA-seq, although it's the most dominant single cell, sequencing technique, it will be used in a lot of studies, but it's not just, as I mentioned, it's not enough to describe the cellular state accurately. And now people realize there's a lot of effort in doing single cell multi-omics. It's basically we're not only want to profile the RNA molecules in the cell, but also we want to profile the DNA molecules, the proteins, the epigenome, spatial coordinate of all the cells. Basically, we want more information, to kind of measure the cellular state from multiple perspectives. So it's like a blind people guessing the elephant, right? If you're touching different place, you might have different conclusion. But if you have more and more measurements over the same cells, cellular states, we will have a better, picture, more accurate picture of the cellular state. So and as I mentioned, because the advantage of the single cell in decoding the cellular dynamics has been used widely study a lot of difficult, long-standing biomedical problems, like in the cancer, right? How to identify and how to deconvolve all the cells, how to identify the critical cell population so that we can design the therapeutics on, or how to like in developmental biology is the same, because at any certain stage, differentiation stage, we always have a heterogeneous cell population. And then if we want to understand how the cell differentiate over time, then we need at least first to deconvolve all the cell population. So except when in this case, in this talk, we're mainly focusing on the cell differentiation applications, like developmental biology applications. And here is an example to describe the, to kind of demonstrate the cellular dynamics in the cell differentiation. Usually is like, the cell will differentiate from all multipotent states, and then, it will differentiate into different determined cellular states. Like one stem cells could differentiate into, blood cells or, epithelial cells, endothelial cells, neuron cells, etc. And now we want to understand how the cell differentiates from multipotent states to different determined states, and more importantly, what dictates the cellular dynamics? What controls the cell fate? And we call this problem as a trajectory inference, and this is a very long-standing problem, and people spend a lot of efforts in building these trajectories, differentiation trajectories or development trajectories in the past. Like in the old days, we don't have single cell measurements. People usually using very labor-intensive type of way, like using microscope. Like John Southern has manually built this trajectory, using the microscope, and he was awarded the Nobel Prize for this contribution. But if we try to using this type of way to build the trajectory, the cell differentiation trajectory, dynamics in complex, advanced species like in human or mouse, that's impossible because we have trillions of cells. It is impossible for us to manually build this trajectory. And single cell genomics is actually providing us opportunities to computationally reconstruct the cellular trajectories, or cellular trajectory to represent the dynamics. And here is one such example. This paper published in 2018 in Science, and the authors in this paper computationally reconstruct the trajectories from a time series single cell measurements. And then we kind of, that's the background. So we kind of formalize the question a bit. It's like we want, giving the single cell measurements on the left side, each dot here represents a cell, for example, and how are we going to build this dynamics trajectory in a matter like in a matter of in this example is like a tree kind of structures. How can we build models that can get the right side from the left side, the single cell measurement, and that's the challenge we want to address in this. So and like here, so many of you may already know that some existing methods can do this, right? Kind of building the trajectory from the single cell measurement, like from the stem cell here, or a multipotent cellular state here, and then the cells can differentiate into 1, 2, 3, 4. 1, 2, 3, 4, 4 different terminal states. And many of these methods, like monoclonal, Siraj, reform, they're able to give, relatively speaking, accurate trajectory in this case, to kind of describe the change over the cellular state over time. But it's not enough for us to manipulate the cell fate, right? Even okay, this can change, from the stem cell here and can differentiate into the orange cells here. But we don't know how we can control the cell fate. How are we gonna, force all the cells to differentiate more into this orange space? So if we did, for example, if this is the lung epithelial cell that we want to kind of repress all other passes, but kind of enhance this part so that we can get more of this cell fate out of this progenitor, right? And existing methods can give us such information. What kind of regulatory, transcription factors or what kind of pathways we should target on, so that we can get more orange cells. And then to fill this gap, I developed a method and called SCDIFF, which was published in Genome Research in 2018. And here actually is we kind of incorporate the gene regulating network as part of the trajectory inference, right? So in high network, you see, all these trajectories or the cellular dynamics are modeled as graph here, right? You have nodes and you have edges, and the nodes here represents the cellular state, or certain cellular state, like or either cell type or cell population, depending on the application scenario. So here you can see, okay, each of these clusters represent our cell type, and then these edges represent the transition or the change between cell types, right? And in here, what we do, an advantage, the uniqueness of our method is we associate our gene regulating network, like GRN here, so that GRN can explain the expression difference or the expression change between states. So it's kind of forcing the model to learn the drivers. So if you can give me this set of TF and the target genes, And based on this set of GF and TF and their target genes, we can predict the expression change. And you know, and the predicted expression, you know, is in other words, the inferred set of, you know, we kind of force us to infer the best set of TF for each of these ages. So that's it. It can explain the expression difference or the expression change between states. So, and this how we build the initial model is very similar to all other existing ones, just entirely based on the gene expression. No extra information is used when we're building the initial model, the initial graph tree. But then we kind of incorporate more information, like, you know, the public TF gene interaction, you know, what TF regulating what gene in the general case. And we also have the expression for all this TF and the genes. And that information, we kind of use that to incorporate that to the model, so that we can iteratively refine this graph, you know, refine these clusters and ages. You know, this age might change, you know, and then we through multiple iterations we will, you know, converge and so yeah. Go ahead if you have questions. So I'll explain this in detail in later stage. So here is how we're building the initial graph. So we start with clustering, you know, this cluster or the cells in each of this time point here. And then, you know, of course, we have to use multiple strategies to learn the best key, you know, model selection type of analysis, and find the best key for each time point. And then we cluster all the cells. You know, in this one, you have one cluster. In this, we have multiple, 3 cluster, 4 cluster. But another thing is we need to synchronize all these clusters, right? If this is a time series analysis, you can capture this in day 1, day 3, day 4. But you know, all the cells captured at the same day, you know, day 1, for example, they're not necessarily at a similar differentiated stage. Some cells may be differentiated faster than others, right? They're more differentiated than others because difference of the cell cycles or others. So now we need to synchronize this. How are we going to synchronize all these clusters to put cells clustered at the similar stage together in the same level? How are we going to do this? There's multiple strategies has been proposed for doing this. So one is using entropy. So a lot of people are doing, you know, synchronize all these cell clusters using entropy. The idea is, okay, if the cell is you know, stem cell stage or undifferentiated stage, you know, the gene expression will be more dynamic, more random, right? Because it's more, you know, kind of unrestricted, right? The expressions can fluctuate a lot between cells, between different stem cells. So along with the differentiation, if the cell become more differentiated, you know, it becomes to a certain long epithelial cell, for example, and then the gene expression will be more restricted. You will have less randomness in the expression. So, and then when you quantify the entropy, you will find a decrease of entropy along with the differentiation. Then based on entropy, you can say, okay, whether this group of cells are more differentiated than this one or this one, you know, or so, and now based on that, we can quantify, we can kind of synchronize all the clusters. And then another way is, you know, as I did here, is it's based on, you know, we can synchronize all these clusters based on the distance to a group of root cells. So in many cases, in the differentiation studies, we can always use in some markers, you know, stem cell markers, for example, right? And then we can use these markers to pick, or, you know, pick some root cells we can say is like the progenitor or undifferentiated cells. And then we can calculate the distance between all other cells to this group of root cells. And so that if you are more differentiated, then the distance will be larger. And if you is less differentiated, then you should be closer to the unrooted cells. And based on this distance, we can put the cells into different levels. Of course, there's some settings here. And once we're doing this, you know, kind of synchronize all these classes, we can build in the initial tree by, you know, connecting the cluster to the closest one in the preceding level. You know, the underlying assumption is, or, you know, the rationale behind this is like the cluster, the parent for these clusters should be more similar to itself compared to all other clusters in the preceding level. So, and this is only the initial graph. So, and then once we get an initial graph, so we will incorporate the TF. Because we want to find the drivers for the cellular transition, and then we will incorporate this TF into this model to iteratively refine the tree, right? And here is, but for that we need to represent each, is in this tree, right? Each node represents a cluster, or that also represent our cellular state, right? Like or cell type. So and then how to represent the cell type here. And here we're using, or for example, we're using a Gaussian to represent, all the expression in this cluster. Of course, because we're using a mixture model, not just a simple Gaussian. But here, for simplicity, we're just using a Gaussian assumption here to to represent the expression in each of these states, in each of these clusters. And here, in other words, we only need two vectors. One is the mu vector, one is the sigma vector. It's like mu and sigma, mean and the standard deviation, to represent the expression in each of all these clusters. And then, and here, because the single cell measurement is very noisy, so for example, it suffers from the dropout, which is because of the limitation of the technology. So a lot of gene expressions are measured as 0, but in reality, it's not 0. The 0 is just a kind of dropout, which is inaccurate. So this could be understood that single cell measurement are really noisy. And then if we really want to get accurate estimation of this expression in each of these states, then we need to find a better strategy. And here we're using a strategy called the common filter. And we borrow this idea from the GPS global navigation system. The reason is the single cell environment in many cases is really similar to the GPS device environment. Like due to the long travel distance to the satellites, right? The GPS device is also suffering, a lot of noise and dropout. For example, if you go into a tunnel and your connection with the satellite was cut off, and then you can't get any readout in that time in that time frame, right? And then it could be understood as a type of dropout. So, and what the GPS device does is like, okay, it not just estimate the position or the location of the device based on the direct readout from the satellite, direct readout, the signals get from the signal, from the satellite, but it also estimate the position of the device, like the car, based on the position of the device in the previous time point, plus the position change. And the position change could be estimated based on the velocity of the car and also the time span, right? So in that case, it can estimate the position of the device from two independent two sources. And here we're doing the same, right? We know the direct measurements of the gene expression is not accurate. It has a lot of noise, especially in dropout. So we don't trust this entirely. And but similarly, if we want to estimate expression in the state S, we can based on the expression in the previous state, like PS, the parent state, and then plus the expression change. So if we know the expression change, and also the expression in the previous state, we can also estimate expression in this state. So in that case, we can also estimate the expression from two different sources, and that will help us to better estimate the true expression in each of the states. And and then with this better estimate, better estimation of this expression, and then we can reassign, because the initial cell assignment are based on clustering. And then once we have a more accurate estimation of the expression for each of these clusters, each of the states, we can reassign the cells back to the states. And then we will iterate between, once we have these reassignment, we can build our updated tree, and then we can iterate between these two steps. Cell reassignment and updating the model until convergency, or maybe your solving criteria is met. So then and in this process, as the expression change was predicted based on owning the TF, what TF are regulating what genes, and the expression of this TF. So we kind of force the model to learn the best set of TF that can dictate the expression change between states. So that's why after we get a model, we will have a best set of drivers, the TF, for self-weight manipulation. So and we apply this method into multiple data set. And one is the long epithelial differentiation data set in this one, and this is a reconstructing model and we actually have we find very the trajectory that we get are consistency with no knowledge, right? Like the ciliated and Clara are more differentiated earlier from the progenitor, and then 81, 82 are differentiated at a later time point. So, and then we also, that's still like a computational validation, right? So we kind of validate the model predictions in this differentiation. For example, in our model we find HIF18, this TF are regulating both, the differentiation of 82 and 81, and also the expression is decreased in this stage. So on the other hand, based on this, we can predict if we, because this TF, the decrease of this TF are causing, are kind of regulating the differentiating of these two cell types, 81 and 82, the alveolar type 1 and type 2. So, and then if we change the trend, if we're not decreasing this expression, if we overexpress this TF, what will happen? So should we will the differentiation of 81 and 82 will be disrupted? So, and then we perform the experiments with collaboration with Jeffrey. And here is the green and red represent this 82 and 81 cell types. And green represents 81, red represent 82. So and this is the bottom one is overexpression, and this is just the normal. So and then you can see if we overexpress this, you significantly disrupt the green and red cells. 81 and 82, the differentiating of 81 and 82 cells. This confirms that this TF is really important in regulating these two cell types. So we also have another study, application of this model in studying the hot differentiating. So and this is the computational model that we get, and then this is the biological replicates of that, and and this is very consistent with no knowledge in hot development. And we also find a novel regulator HOPX from the model. And if we overexpress this HOPX, the model, as predicted here, is really affect the maturity of the chimera-cells, which is hard muscle cells. This is another confirmation, experimental validation of our model. So to summarize, this, if we the model can really discover novel regulators, right? And by targeting on these regulators, we can really change the cell fate, right? For example, if we overexpress this TF, we can enhance the maturation of chimera-cells. If we lockout this, we will inhibit the maturation of chimera-cells, because you can't get mature chimera-cells if we lockout this. Or it's more difficult to get mature chimera-cells if we lockout this TF. So this is suggesting us, the model can really give us, regulators that could be used for self-weight manipulation. So, and that's the first study. And then we have another one. I want to just, I want to speed up a bit. So it's another application, is still, we want to, in this case, we want to, is collaboration with direct coding. So in this case, we want to get So for line regenerative medicine, we want to from induce the stem cell, induce the iPSC, induce the stem cells. We want to get more lung epithelial cells. But the conventional differentiation protocol is really ineffective. So usually we get less than 5% of lung epithelial cells. What we want, the kind of design cell from the iPSC using the standard way. So and we want to understand. So based on the knowledge, prior knowledge from Darrow, we know the critical time window is after day 15 of this reprogramming. From day 15 to day 31. But we don't actually know what are the critical time points here. And we can't afford to do single cell measurement for 15 days, 16 days in total, because that will be very expensive. But on the other hand, maybe we don't need actually to provide the single cell every day or every 2 days, because not all these time points are equally important. There's always some time points are more critical. Namely, some time points may not that important. So then the answer is, how do we identify the most critical time points so we can spend the money on those critical time points to provide the single cell measurement, right? Kind of maximize the budget, the information that's being gathered with the budget. So the solution we find is we will use the bulk measurement, which is very affordable, which is very compared to single cell is almost nothing in terms of the cost. So we select a lot of markers, like here, like over 80 different markers about the the differentiation of the lung epithelial cells, and then this is the actual expression, and then we try to select the time points. And here you have 9 different time points, right? And then so can we select a fewer number of time points, like 4 or 5, but we still, based on this choosing time points, we can reconstruct the expression accurately. And then right? So and then now is if we select more time points, the reconstruction error will be lower because you have more information about the expression, right? And on the other hand, if we reduce the number of time points that we want to profile, we will have an error. We have increase the reconstruction error. Reconstruction error means the actual expression that we measured. The difference between the actual measured expression versus reconstructed expression from the selected time point. Like if you select only these 2 time points, and based on only these 2, fit a spline curve, and then based on this fit a spline curve to estimate expression for other time points, and then you compare to the original one, you calculate or error. And actually, we now based on this curve, we can find out a best trade-off point here. The reconstruction error is low, but also you don't have to provide single cell for all time points, like all 9 time points. And we'll also have a bottom line here, which is the repeat error, which means the it's the difference between replicates. You can't go beyond that, right? If even you provide all time points, you will still have a reconstruction error because you have difference between replicates. So that's the best. So if you can see here, if you're only using 6 time point, the reconstruction error is already close enough to the the best you can get. So and based on that, we can for example, in this case, if we select 6 time point, we reconstruct all this expression, and this reconstructed expression is really close to the original measurement. If you look at if you compare that. So and based on that, based on this kind of for more affordable bulk analysis, we can select the best time point in this differentiation study. And here we select 6, and then we measure all this time, cells from the 6 time points. And now we kind of build using or model improved version of SCDIFF as I discussed, because the SCDIFF is discrete. Here we kind of extending to continuous the model, and then here is and then we apply the method on the time series single cell measurement from the 6 time points, and then we get different, kind of branches, and we find, okay, the top branch, the lower branches are all like NKX21 positive, and this is the marker for lung epithelial cells in general. All right, and now we find the top passes actually is NKX2 is not lung epithelial, is not lung cells in general, is actually is intestinal cells. And we also find, okay, the Wnt signaling regulators are kind of more, the expression are significantly high in the top passes. So which is suggesting, okay, the Wnt signaling might be, is definitely higher in the top versus bottom. And which is suggesting that, okay, Wnt signaling might be the key, the causing the split, right? And also from this model, we can estimate this actual time of the split. And then based on our model estimation, the split time is 17.5 around that day. So, and okay, this model is telling us, okay, Wnt signaling might causing the divergence of non versus non cells. And the split happened around day 17. So this is the model prediction. And then, okay, I will skip this part because this, is another validation of that. But we have another, set of validation I want to talk about. So, this actually is showing, is really this Wnt markers is really different, right? In this one and this one, this is Wnt signaling repressor, so it's higher in the bottom one. So, and this is the marker for the Wnt signaling is higher in the top passes. So it's really showing this Wnt signaling relevant, markers are really differential, are really, different between the top and versus bottom passes. The top are the Wnt signaling high, the bottom are Wnt signaling low. So because we are more interested in getting lung epithelial cells like bottom passes, so we want to kind of repress the top passes. And based on that, we need to target on the Wnt signaling. And then we perform the experiment, the child withdrawal, which is a way to repress the Wnt signaling. And also we did it at different time points, right? Day 17, day 19. And is that just as model predicted, if we repress the Wnt signaling at day 17, here is the general lung epithelial cells. This is one specific 82 lung epithelial cells, which did, or kind of subtypes of the nerves. If you can look at the production and how many stem cells could be differentiated into this cell type, and you can see with, if we repress the Wnt signaling at day 17, we double the production, from less than 5% to over 10%. So and this is actually, or significant advancement of the or improving non-epithelial or differentiating non-epithelial from stem cells, and this work was published in Cell Stem Cell. So, and so to summarize, so we can, we can, based on the single cell genomics, right? Based on the single cell environment, we can really find novel regulators or pathways that could be used to manipulate the cell fate. As I showed here, we doubled the production of lung epithelial cells, 82, right? And another one is large-scale single-cell studies require really careful planning, about when and how to sample. So imagine if we don't, if we don't capture this critical at day 17, right? And then we might miss the signal at all. So, and then selecting day 17 will be critical, right? In this case. So, and then, and it seems the bulk analysis can really help us to find the critical time window. So if we don't do this, we just manually select this, we might miss this, right? When you choose 15, 16, and 18, you might miss the critical time window. So that's why when we're doing this kind of single cell environment, especially in time series single cell environment, we really need to carefully select the time points. We don't want to waste our money on, less informative time points. So yeah. So, and then lastly, repressing Wnt signaling can really increase the production of lung epithelial cells. So, and that's it. Any questions?
Thank you so much. It's really a wonderful tour of the single cell world and the challenges involved there. There's a question from Benjamin Smith, or Donald Smith. Go ahead.
Yeah. Hi, thanks. Thanks, Jen. That was a nice talk, such a fascinating field. I had a question about, early on you depicted sort of cell differentiation as a cluster of a certain color of cells, and they can have one of 5 fates. And obviously you're exploring what the gene expression states are of each of those and the transitions and so on. You mentioned that in your approach, this notion of entropy, and that the less differentiated or more pluripotent state has higher entropy. And then you also depicted differentiation as unidirectional arrows. My question is there evidence to support those? Well, one, are those critical assumptions to your method? And 2, is there evidence to support that a more undifferentiated cell is in somehow more disordered, or not? And also, are the arrows unidirectional, or does it require sort of experimental perturbation to get it to go in the opposite direction?
So yeah, there's like synchronize all these cells. Sorry. And here, yes. So yeah, there's indeed 2 ways to calculate, to kind of synchronize the stage of all the cells. One is as the entropy, as I discussed. So there's evidence, you can search a lot of public, publications. They are suggesting, okay, stem cells, they have more randomness in the expression. If the cell, you look at the epigenome, right, it's less restricted. The CDCF are more, you have more less clips on there. So it's because the epigenome is more, I would say, freedom, is more flexible at the early stage. So that's, the expression is more dynamic, more randomness. If we calculate entropy, it is higher compared to more, differentiated. So the epigenome is more, you have more isolation, right, etc. So your epigenome is more constrained. And then the expression is more kind of more determined to some extent. So you quantify the entropy, you do see the difference along with differentiation. But in reality, in practical applications, we find as, if we directly calculate the distance, especially if we have a good set of markers to select the undifferentiated cells, the distance might be a better way, because I tried both strategies in this case. And we find the distance based, based on selecting markers to calculate this, actually performs better. You can, because we know the ground choose for these different cell types, right? We know what is dry, what is not. So, and entropy, in general, they're all very good, but in this case, the distance based is better in many cases. So what's your second part of your question?
Just the direction out, is it do we believe that cells sort of differentiate in a unidirectional sense, or can they change their mind and regress to a more de-differentiated state and go down another pathway?
So in the differentiation studies, it is, but in many cases, there's also, for example, reprogramming studies, it might not be that, because you might reprogram, you change the cellular state to more like stemness, and then you will differentiate after that. But if the direction is from stem cells to more differentiated cells, then yeah, I think so. I think we can quantify, we can use entropy to represent the, use as an arrow to represent the direction of the differentiation.
There's a couple of questions in the chat, June, as well. I don't know if Benjamin, you had additional comments here.
No, I can speak with them offline. Thanks.
Great, thank you. Yue Li, is there any, algorithmically, is there any connection between SCDIFF and module network by Iran Segal or Geronimo by Suin Lee?
No, and I don't know when they published the paper, but we actually developed SCDIFF, quite independently. We don't borrow the ideas from other module network from. We're not, because but we do use, we kind of utilize the existing TF-gene interactions, from both ENCODE or others. So we use that as a starting point, and then we kind of try to find the set of TF genes to explain the expression change for each of these edges in the graph.
Mm-hmm.
So it's not an inference of GRN. It's based on existing GRN. We find the active part of that GRN to explain each of these edges. So it's not the same. I would say it's not the same task. I would say.
Oh yeah, maybe I was reading it a little bit differently. So because I thought that when I look at that iterative process, when you have the markers like TF or separate the samples, and then you try to infer a driver at the same time and try to go back to refine the clusters. So there are some sort of analogy, or if you will, like some similarity. I'm just wondering, like, is there, whether there is connection, but it looks like it's very completely different idea, independent idea. Yeah, but I guess the fact that you're using time series is also different from their model where they don't actually operate on the time series data.
Yeah. So I think that their method may be more like a de novo discovery of GRN, and ours is based on prior knowledge of existing GRN, and then we combine it with the graph, and then to find the most active part of this GRN, because not the general GRN may not be also different component of the GRN may function at different stages. And we need to really find the critical components, and then so that we can target on that to change the cell fate. So
Okay.
yeah.
Thank you.
So we have one more question in the chat, and then we'll go to Dylan after that. From Abhi Kumaran, how can we acquire time series single cell data from difficult to access patient-derived tumors? Will in vitro models capture in vivo patient dynamics?
Yeah.
Just an easy question for you.
This is not so we actually have apply this to study the lung fibrosis. So and then in that case actually we have because in the lung fibrosis in many cases there are already different regions of the lung, they have different extent of fibrosis. So that's kind of spatial diversity represents some temporal diversity. Or so I'm not sure whether that's in your studies, whether there's the cells that you get from different regions of tissue may also represent such a difference in terms of time. And if that's the case, then you can always try to build a pseudo temporal data set from the same tissue. Even you cannot continuously track the cells from the same tissue over time. But even from the same time, there already exist the difference at different stages, right? And then you can use that to build our pseudo time series data set and use it to model.
Thanks. Okay, Dylan.
Yes. Hello, Professor Ding. Can you hear me?
Yeah. Yes.
Okay, so I don't know if you're aware of this theory, which states that cells and aging organs in mammals actually lose their cell identity. So they kind of refer back to some sort of undifferentiated state partially. If that's the case, do you think we could use your analysis to identify the signaling pathways that we require to sort of constrain the aging process?
Yeah, that's a limitation of the model, because here we cannot assign the way we define, we cannot assign the cells to different stages based on the distance to the. We cannot assume the differentiation is the one directional, is go one way. But in your case, you say, okay, you might back and forth, and then it's not a. it's still, we can still use the model, but you need to refine how you're gonna assign how you're gonna synchronize all the cells in a different way. Can you find other features that can represent different stages in your process?
Right. So related to like a previous question about, I think it was a previous question about the relationship, like the directionality of these differentiation process and perhaps entropy related to gene expression. Is it possible to understand these processes in terms of the bidirectionality of the potential differentiation process? So you're just saying how the limitation is only going the forward direction, but the way I understood it from your presentation is it's not necessarily a huge leap to go in the backwards process if you've established the differentiation tree structure, really. What's your thoughts on that? Like going backwards, is that really an issue if you already have the trajectories mapped out? Or?
So if that's one directional, it's easy to infer the parents, right? Because you will build a tree, you need parents for each node. But if it's going back and forth, how are you gonna know whether the parent-children relationship between two?
Meaning if you already had your tree structure mapped out, right?
But how do you build the initial tree? You need to rely on something, right?
But if you had, I mean, I guess I'm being very like general and high level thinking here. Like if you had that mapped out as maybe as an atlas or sort of as a reference set of trajectory trees, they're already mapped out. You just understand how different signaling pathways that sort of irrigate the flow of differentiation across that structure. I don't know if that was clear, but
yeah, if that's the case, if you say, okay, you have the backbone of this at different stages, you have the tree, and then you can use the model to find the TFs, and also the targeted genes that can explain the expression difference or expression change between states. That's durable. That's that's able. The model can be used for such purpose.
Okay, great. Thanks.
Sorry about that. Are there other questions? I was curious about a little bit some of the models you had talked. Obviously you're assigning the cells to clusters. Is this a probabilistic assignment? Could a cell actually belong to more than one cluster with certain probabilities?
Theoretically speaking, yes, it is a probabilistic assignment. But now we just assign to the state with the maximum probability. But it is, it is a distribution over all the states. So you have 10% chance in this state, 50% chance in this state. So if you want to apply another way, is there. But in this case, we just assign to the state with the maximum probability.
Okay. We are reaching the top of the hour. Are there other questions for Dr. Ding? All right. In that case, I would like to certainly express a thanks for starting off our seminar series for 2022 and doing such a great job of explaining the complexities of single cell trajectory analysis. This was wonderful. Thank you so much.
Thank you for the invitation.